I'm David Rabson from the Advanced Scientific Computing Research Program, and I'm pleased to introduce our first speaker, Mary LaPorte from the University of California, Davis, in plant biology, and she will be telling us about learning from kernels, HPC in plant breeding. So please welcome Mary Frances LaPorte.
Alrighty. Well, hello everyone. I am just ecstatic to be here and chatting with you all today. We'll be talking about learning from kernels, so a little play on words between the corn kernels we'll be talking about and the mathematical concept. And my ultimate goal for the talk today is that every one of us comes away with a greater appreciation of the plants in our life, regardless of what background you're coming from, and also an appreciation for how plant breeders have always been using the latest technology to advance their crops, and HPC is no exception. So, to get started, we just shared a wonderful lunch together. So I thought I'd pose the question, how many plants have you eaten today? So we all just shared a nice mixed salad with some pumpkin seeds and grape tomatoes. If you had the chicken entree, maybe you had some butternut squash. And perhaps you ate a granola bar with some high fructose corn syrup this morning in it, or maybe, I don't know, you had a cob of corn. Well, maybe this will help you think about the fact that each one of these crops that we ate, each one of these foods, didn't just appear one day. They were the result of a lot of effort and a lot of labor over unnumbered generations. So in this meal, we shared these particular foods, but each of them, we know something about their wild relatives. So in biology, we call these wild progenitors. You can think of them as kind of ancestors of a particular plant that we know, a related plant that's alive today. And for example, for our mixed green salad, a wild progenitor of that might be this Lactuca genus, which is still around today, that has these ribbed spines, or these spines on their ribs. And when we were eating our salad, we weren't worried about that. And that's because over a number of generations, people worked through and they selected the lettuce that didn't have these ribs and therefore improved them and made them more tasty. And the same can be true for different squashes. So over the years, plant breeders have bred for different sizes and shapes of squash, different sizes of seeds, etc. This wild progenitor of tomato is like pea-sized and has a completely different flavor than the grape tomatoes we ate today that were introduced to the US in the 1990s. And one of my favorite examples, and I think really charismatic, is the wild progenitor of maize, which is teosinte. And teosinte has these really hard seeds that are like rock hard, and there's only maybe 8 per cob. And we see so many more seeds per cob today on the sweet corn that's really succulent that we eat, or that goes into animal feed. So each of these crops was developed over thousands of years, and they were the product of human selection. So humans selected for these more nutritious, more delicious crops, and they were looking at which crops had the best flavor, the best vigor, perhaps yield, color, nutritional value, disease resistance, climate resilience is a huge thing people are talking about. But perhaps you guys have already heard about this from this XKCD comic that I've heard come up quite a few times at program review over the years. So in it, this main character here talks about, I used to think that Brussels sprouts were terrible, and now I love them. And then their long-haired friend says, that's because people have been working really hard to make that happen. It wasn't an accident. So they make a few more little jokes about that. So one example of this selection over time that I think is quite jaw-dropping is the improvement in yield in maize over the years. So in the 1860s through like the 1940s, the yield per acre of maize stayed about the same. And it wasn't until the advancement of hybrid corn that we start to see this huge increase to the yields that we see today. If you're driving through the Midwest, you're probably familiar with this corn that's grown really close together. It's super tall and doesn't blow over easily in the wind. And that's thanks to the development of hybrid corn, which was a molecular advancement. But this is not the only advancement that has led to this great increase in yield. For example, in like 1950, when we had the advent of the mixed model, that was a huge deal for the development of corn. And then also kind of in this time after this 100% acceptance or adoption of hybrid corn, we have the development of best linear unbiased predictors. We're seeing that in the literature, and corn science has never been the same. Like that's the way that you have to calculate any sort of field trial results. And so these advancements in computation and mathematics go hand in hand with these advancements in corn. And I've included this image of a key punch machine to remember to talk about. One of the people on my dissertation committee always talks about how when he was doing his PhD, carrying his box full of punch cards over to the computing center, and the fact we don't have to do that now. So all of this is to say that these improvements in crop sciences go hand in hand with improvements in computational sciences, and this is not the very beginning of us using the most advanced computation in the moment. So now let's get into my research. So the ultimate goal of my research is biofortification. And biofortification means to increase the nutritional value of a particular crop. So instead of selecting for yield, we're selecting for nutritional quality for people. And so generally in biofortification, we're talking about improving nutritional quality, specifically through breeding and agronomic practices, and not through gene editing, like CRISPR, for example. Although CRISPR is super important for plant breeders, and plant breeders use it all the time. With biofortification, the general goal is to get a crop deployed as quickly as possible, and that's just not always possible with gene editing in every country around the world. So it's important to keep in mind the political context. So specifically, we're looking at biofortifying for a compound or a set of compounds called carotenoids. And so carotenoids live in the maize grain, or the kernel is another way to put it, as you can see here. So I've highlighted it, but there's lots of other things you could select for in biofortification, which is defined in this paper here. And so carotenoids, the body can synthesize into vitamin A. We can't make vitamin A naturally. We have to get it through our diet. And so many people over the last, especially 10 years, have developed or have been working on developing biofortified corn kernels or maize that has a very high concentration of these carotenoids in the kernels. And so this is the product of some of that labor by different academic institutions, NGOs, et cetera. So in the back here, we have a corn dish that looks kind of white in color, and that comes from a white maize. And then in the front, we have a yellowish that comes from a biofortified orange maize grain. But the thing is, despite all of these advancements, there's still room for more improvement because carotenoids degrade over time. So it's important to have as high of a concentration as possible when it gets to the consumer. So we are looking to increase the concentration of maize grain carotenoids. And to do that, you have to make selections. We talked about selections earlier, like getting the spines out of lettuce. So we're doing this in real time. Plant breeders are doing this in real time. But we have more tools available to us now in order to make these selections. So in particular, I'll tell you about one tool that we use called genomic prediction to make better selections. So genomic prediction is using genetic markers, so genetic markers scattered throughout the genome, as predictors to predict a particular phenotype. And that allows for people to make selections more rapidly. So if you're a breeder, you make 10,000 crosses, you do not have time to go in the field and look at and quantify carotenoids in 10,000 different plants. You just don't. So you need a way to sift through that information, take perhaps the top half or just get rid of the worst ones. And then from there, you can do your quantification and move forward. So let me describe genomic prediction practically. So you have a training population of some sort of your corn, your maize of interest, and from there, you take the phenotypes. So for example, for us, it could be maize grain carotenoids. You genotype it, so you get that sequence of markers throughout the genome, and then you fit some sort of model where you're predicting your phenotype based on your genotype. And then from there, what you can do is you'll have this model, and you can just take the genotypic information and plug it into your model and estimate, get a y-hat for your for a new set of maize plants that you don't know their y value for. So that's kind of the ultimate goal in selection. But if you want to see how well your model is doing, what you can do is take a population where you have everything or almost everything, both genotyped and phenotyped. You can fit your model, and then you can for lines that you know what their phenotype will be, you can create your model and then estimate and compare your y hat, do a correlation between your y hat and your known value for y, and find predictive ability. So that's the kind of results I'll be telling you about in a moment. In this particular case, the phenotype I'm thinking about is vitamin A-related traits in a kernel. So here are some examples of the biosynthetic pathway for maize grain carotenoids. We have the genotypes for this plant, and we're trying a variety of models for genomic prediction. So this is the type of results I'll be showing you. But before I show you those, let me tell you a little bit about how we grew the maize. So this maize was grown in 2 different sites in central Mexico, Agua Fria and Tlaxapan, from the years 2010 through 2013. And this maize target population was designed specifically to look at carotenoids. And they were quantified for these particular compounds. So I'll talk about lutein, beta-carotene, beta-cryptozanthin, and zeaxanthin. And so I show you this biosynthetic pathway again to emphasize the fact that there are known genes that we know are related to these traits of interest. So we were interested in looking at genomic prediction in this population that was grown in Mexico. So here is a little bit of results. So we compared the predictive ability, again, that correlation between y hat and y, for several different models, and I'm just highlighting 3 of them here. On the y-axis, we have that correlation, the predictive ability, and then on the x-axis, we have which environment it was grown in, so Agua Fria or Tlaxapan. And one thing we were curious about, since we know that there are a set of genes that are related to these phenotypes of interest, will a method that uses all of the markers scattered throughout the genome, for example, RR-BLEP, which is kind of analogous to ridge regression with a component for the population structure, would this method perform better or worse than a method like LASSO that does feature selection? Perhaps because we already know that there are a few genes related, a method with feature selection will do better. Or will reproducing kernel Hilbert spaces, there's a kernel again, do better since we know there are quite a few genes that are related to these traits of interest. So we compared them all, and what we found was in most traits, each one of these panels is a trait. In most traits and in most environments, RR-BLEP, the method that used all markers, actually performed the best. RR-BLEP is a workhorse for genomic prediction, so we weren't necessarily surprised about that. That was pretty consistent with many traits in the literature. But then we were curious, okay, well, specifically, carotenoid traits have a kind of middle number of genes identified, like maybe in the order of dozens. We call that an oligogenic trait, or perhaps a relatively oligogenic trait in the genetics world. So would it do better to just use those traits that are the markers that are in proximity of the genes related to our traits of interest, or should we use all of the markers spread throughout the genome to do genomic prediction? So to do this, we were interested in comparing a 2-gene approach, where we looked at the markers that were just in proximity to a few genes of interest, versus RR-BLEP, which uses all of the markers. So in this case, that's like 550K. It's a lot of markers versus just the ones in proximity to these 2 genes of interest or 13 genes of interest. And what we found, very surprisingly, was that, again, in most traits and in most environments, the predictive ability was the highest for RR-BLEP, meaning that there's some advantage conferred to predictive ability when you include all of the markers in the genome for this relatively oligogenic trait versus these methods like 2-gene selection, which tend to do a bit worse. In particular, they're better for, RR-BLEP is better than the 2-gene approach for these traits for which we have markers that aren't necessarily the best for lutein and zeaxanthin. So for many carotenoid traits, RR-BLEP is much better. And then finally, what I'll tell you about, you might be thinking, okay, this was in 2010, so how do you know that this is relevant to 2011 or the next year? So we wanted to look and see, do our predictions hold between years? If I create my model for 1 year, can I use it to predict the value for the next year, the breeding value for the next year? So in this case, on the y-axis, again, we have predictive ability. We have the traits on the x-axis, and then sets of environments for each pane. So for each of these blue violins, the model was trained in one environment and tested in the same environment. And for the green violins to the right, they were trained in one environment and tested in a second environment. So the main takeaway from this is, for the most part, this looks like a straight line, which means that the model performs just about as well in the second environment as it does in the first, meaning that we can use our models to predict into a different year. Notable exception is lutein. So lutein was quantified differently with a different platform in Agua Fria than it was in Tlatzepán, which also shows that you have to be careful about which platforms you're quantifying in if you'd like to be able to predict into a future year, select for a different year. So the main takeaways from this talk are that crops are a technology in general. The worlds of plant breeding and plant sciences, HPC is both critical and it's very widespread throughout our field. Biologically, the biggest finding from this was that predictive ability was actually higher for whole genome prediction than it was for prediction that only included genes of interest. And practically, if you're going to be doing one of these types of experiment, RBLEP's great, and it's good to measure things consistently. So thank you. I wanted to take a moment to thank my, my lab group, especially my PI, Christine here, the CSGF for making graduate school possible for me and for introducing me to all of you. I'm eternally grateful. And then also to my family, especially Emily, Samuel, and Teresa, who took the train up here today. And if you want to check out these results, you can check it out here. So yeah, what questions do you have?